Hello everyone. This is the lecture for biological molecules, and we will be starting off by talking about carbon compounds, and then we'll get into familiar territory where we start talking about things like proteins and lipids, nucleic acids, and carbohydrates. So from a structural and building life perspective, carbon is a key component, it's an essential element, and it's because of carbon's behavior. So carbon forms the backbone of all of your biological molecules. We're going to go ahead and highlight that because that's really important. Now, carbon can be in the form of chains where the carbon molecules are linked up to each other, or they can be in a form of a ring where they're almost circular looking, and they make up the backbones of all of your biological molecules. So what are the biological molecules? Because you're going to hear these terms. You're going to hear this term a lot, but also we have other terms that are interchangeable but mean the same thing. So organic molecules are anything that's made or based in carbon. Carbon forms the base of the molecule itself. We also refer to them as biomolecules, organic molecules, like I've mentioned, biological molecules, or sometimes we call them macromolecules because they are large. All of them are referring to the molecules that use carbon as their base element in their composition. So why is carbon so key? It's because carbon has the unique ability to form 4 bonds. If you think back to the periodic table, carbon is in group 4A, which means that it has 4 valence electrons. Now remember, every element, when it gets involved in a chemical reaction, is trying to become stable. And to be stable, I need to have 8 electrons in my valence shell. So carbon is in the unique position of I can add 4 more, or I can lose the 4 that I already have. So it makes it fairly reactive, and it also means that carbon can form 4 different bonds. Being able to form these 4 different bonds allows carbon to take on a variety of shapes, a variety of appearances. So carbon on its own, bonding carbon to carbon, can form single bonds, double bonds, or even triple bonds, and pictures of those are off to the side. So here's my carbon molecule. Just focus on the carbon for now. Here's my carbon molecules. Notice they are bonded to each other, so they're making up the base. So that's 1 bond. I'm going to number these for you. So this is bond number 1. This would be bond number 2, number 3, and number 4 for just that first carbon molecule. For the second carbon molecule, that middle bond is still bond number 1, followed by 2, followed by 3, followed by 4. Each carbon molecule is capable of bonding 4 times. So that's what a single bond looks like. With a double bond, so the molecule next to it, the carbon-to-carbon bond the carbon-to-carbon molecules have 2 bonds between them. So bond 1 and bond 2. That makes up my double bond. But carbon can still bond 2 more times. So here we are 3 and 4 for the carbon on the left, 3 and 4 for the carbon on the right. And then finally, carbon can form triple bonds with itself. So bond 1, bond 2, bond 3, all in between the 2 carbon molecules. There is still 1 bond available for each of these carbons. So the carbon on the left can bond here for a 4th bond. The carbon on the right can bond here for a 4th bond. So that's how we get single, double, and triple bonds. Now these would be examples of chains because they are next to each other, connected, linked up, like the links in a chain. Versus this picture down here would be an example of a ring. So instead of the carbons being lined up next to each other, they connect to the carbons in front of them and behind them to form a hexagonal, circular-ish type shape. Really, it's hexagonal, but. So you would notice that on those pictures we just looked at, those carbons had hydrogens bonded to them. When hydrogens and carbons bond to each other, we call those hydrocarbons. Now, hydrogens aren't the only thing that carbons can bond, but you will see hydrogens bonded to carbons a lot, okay? So just know that carbon has the ability to covalently bond to each other, and then they can also bond to a bunch of other kinds of molecules that can be just a few atoms big to thousands or millions or, tens of millions, if you will, atoms big. And that's really important because that's what's going to give each of our biomolecules its own specific identity. So we have a picture here of methane. Methane is an example of the simplest kind of hydrocarbon. It is one carbon molecule bonded to 4 hydrogen molecules. Okay, and all these bonds, would be covalent because this way carbon gets its 8 bonds because it's sharing the electrons with hydrogen, and hydrogens get their 2 electrons because they are sharing their 1 electron with the one that they're bonded to in the carbon. Throwing in a little chemistry review there for you. Okay, so here are a few more examples of some simple carbon molecules. We have ethane, which is 2 carbon molecules that are then also bonded to 6 hydrogen molecules. And then we have another example is propane, where we'd have 3 carbon molecules. Butane, we'd have 4 carbon molecules, and it just keeps going from there. As long as you remember that carbon bonds 4 times, then for the most part, how many hydrogens it can accommodate would make sense. As I pointed out a couple of slides ago, they can also form these ring structures. So this is another example of the ring structure. And then this is an example of the chain structure, where they're all next to each other. Boom, 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 boom. So when living things die, or when they produce waste, and it gets broken down into its simplest components, hydrocarbons are normally what it gets broken down into. So let's talk about the actual macromolecules now. So the word macro means large. Sorry, switching my highlighter. And macromolecules are large organic molecules. So large meaning they're big, they take up a lot of space. Organic tells me that they're based in carbon. And these macromolecules are found in all living organisms. They contain carbon and hydrogen plus other elements. So there are 4 classes of macromolecules, and they are the carbohydrates, or carbs, the lipids, which we sometimes call fats, proteins, which you're probably very familiar with, and your nucleic acids, or your genetic material. All right, so just to add in those more common terms to help you attach this new knowledge to things that you are already familiar with. So what are these other molecules that the macromolecules can have attached to them? So we can have small reactive groups of atoms that anchor themselves to the organic molecules in different combinations and in different locations. We call them functional groups. And they tend to be the part of the macromolecule that's actively involved in a reaction. So they are the reactive sections, if that makes sense. Okay? They have covalent bonds, but their covalent bonds are more easily broken or rearranged than in any other part of the molecule. So your covalent bonds that are holding your carbons to your carbons are going to be the most strong and the most solid, but the ones that are involved in these functional groups will be more easy to manipulate because that's the part of the molecule that's going to take part in any chemical activity. So it's really important for you to note here that the hydrocarbon part of the macromolecule does not engage in chemical reactions. It's just the functional group that's going to be a part of any chemical reactions. Okay, so now let's talk about some specific functional groups, and you do need to be able to recognize these and name these if they are a part of a molecule. So I know that my image looks different to the one on your PowerPoint. It did not transfer for some reason into my program for making the video, but the information will be the same. though. Don't stress. So the simplest and the first functional group we're going to talk about are the hydroxyls. These are the OHs. There's another way to think of them. And you can just highlight the portions of this on your sheet that I reference. You probably don't need to write in a lot, because a lot of the things I will write in are things that already exist on your pages. So the most important part of the molecule, the part that's actually the functional group, is the oxygen bonded to the hydrogen, okay? We commonly refer to them as the alcohols. So that's something you would want to highlight. Excuse me. And in terms of their properties, they are polar. So that just means that, that is not a highlighter. There we go. So they are polar molecules, which means that they can hydrogen bond with water. So if you put them in a water solution, they will hydrogen bond with the water molecules. Okay, the next group is known as the carbonyls. And in terms of what they look like, they are going to be a carbon double bonded to an oxygen. These actually come in two forms, and depending on where the molecule is found, they are called different things. So if the molecule is found at the end of the macromolecule, they are known as aldehydes. So that just means that the functional group is found at the end. Okay, and if you look at your image, that top one, you can see what I mean. So we have two carbons making up this molecule or this compound, and the C double bonded to the O is on the end carbon. So it's called a carbonyl. And know that it can be on either end, on the left end or the right end. As long as it's on the very last or very first carbon, it is an aldehyde. Now I'm going to have to draw in the next one. You, again, already have it drawn, but I forgot to crop that part of my picture. So another way that these can appear is in the form of a ketone. So with ketones, the functional region is going to be found in the middle. So pretty much not at the end is the way you want to think about that, okay? In terms of properties, they too are going to be polar, and you will see these a lot in our carbs, okay? You find carbonyl features or carbonyl functional groups a lot in our carbohydrate type macromolecules. The next two groups we're going to talk about are carboxyl groups and amino groups. So carboxylic acids or carboxyls are a type of organic acid. Sorry, I forgot the A. So remember, when we say acid, we're just talking about a substance that is capable of releasing hydrogen ions. Again, I'm going to tie in past topics as much as I can every time we do a video, so that way you have a quick little review here and there as we go. So with my carboxylic acid, the functional part of the carboxylic acid is the carbon that is bonded to a double bond to an oxygen and a single bond to a hydroxyl group. Now, this image shows what we call chemistry shorthand. So where these two lines meet, there's technically a carbon there. They don't draw it in because if you've been doing science for a while, you just know that. That's something that you're taught. But just to make sure everybody's on the same page, there's technically a carbon there. There's technically a carbon here. So the important part, the functional group part, if you will, is this whole region. Okay, it's that COOH. But one of those O's is double bonded, and then the other one is a hydroxyl, so it's already attached to a hydrogen. In terms of a great example of a carboxyl acid, that would be like vinegar, acetic acid, okay? And because of that OH group, they are able to release hydrogen. And that's why they're considered an acid. So it means that they would be reactive in water. Now, the next group is called the amino group. You may also see it referred to as the amines. And if you're familiar with the term amino acid, then the amino group is why amino acids have that word in their name. So their functional group is actually a nitrogen, not a carbon, that is bonded to two hydrogens, one on either side. So we call this an NH2 group. And all of our amino acids have this NH2 group as part of its base. So these guys have the ability to accept hydrogen, so they act more like a base. They are not going to behave like an acid. Okay, so the next two functional groups are the phosphate group and the sulfhydride group. Your phosphate group is going to be a phosphate that's bonded to let me rephrase that. A phosphate that's double bonded to an oxygen, single bonded to another oxygen, and then single bonded to two more hydroxyl groups. So completely surrounded by oxygens, let's say. So because of all the oxygens, it's going to be charged. They are going to be able to release hydrogens, so there will be some reactions taking place. They're capable of being reactive. You're going to see phosphate groups a lot on your nucleic acids. Then next we have the sulfhydride, and that is just a sulfur that's bonded to a hydrogen. These guys are polar, so they will form hydrogen bonds in water. They also have the ability to link to other sulfhydride groups and form what's known as a disulfide linkage or a disulfide bridge. Okay, and then the last functional group for this little section is going to be the methyl group. Your methyl group is a very basic functional group. It's just carbon bonded to three hydrogens. So like this section of the molecule would be methyl. It doesn't have any like super specific properties, but it does exist, so we need to talk about it. So not all functional groups are created equally in terms of reactivity. Some of them are more reactive than others. So this is just a general hierarchy of the order, if you will, of most reactive to least reactive. Okay, so first and foremost, charged functional groups, so the ones with ions such as phosphate, are always going to be the most reactive. The more ions they have, the more likely those ions are going to want to bond to make themselves stable. Functional groups with double bonds, so the carboxyls, the aldehydes, the ketones, they're all going to want to react. Functional groups with single bonds, such as your alcohols, they're going to be third in the list. And then methyl, which is that C bonded to your three H's, they're already, for the most part, close to being super stable. Those hydrogens are really stable because they're already bonded. Carbon's already bonded to another carbon, so they're going to be the least reactive of all. So because compounds are made up of atoms that can be arranged in certain ways, let's say, they have the ability to form what are known as isomers. So isomers have the same chemical formula, but they're put together in a different way. So molecularly, their structures can look very different. And there are two types of isomers. So there's stereoisomers, which form mirror images. So when you put the two next to each other, it's like looking in a mirror. They're reflected against each other. And then we have structural isomers, which are two molecules that have the same chemical formula, but again, the atoms are arranged in different ways, in unique ways. And again, sometimes the image doesn't translate properly, but it should be fine on your slide. So this should be C double bond O, and then here should be another C double bond O. And it's showing how this is a structural isomer. So on this version of glucose, the C double bond O, the carboxylic acid, is on the end. Or, sorry, not the carboxylic acid, sorry, the aldehyde, sorry, is on the end. But in fructose, the difference is that the C double bond O is in the middle. It's not on the end, so it's now a ketone. So this one is an aldehyde versus a ketone. Everything else is the same. If we counted all the hydrogens and oxygens and carbons, we get the exact same number. The difference is just where did the double bond O go. Now, specifically when the arrangement involves a carbon molecule, these structures are known as enantiomers. So the carbon, like one version of the molecule versus the other version of the molecule, what's on that carbon gets flipped around. So if you look at the image here, so here's my carbon, and on diglyceraldehyde, I have my hydroxide on the right and my hydrogen on the left. But on L-glyceraldehyde, it's flopped around. So think of that carbon as twisting. And now my OH group is going to be on the left and my hydrogen is on the right. So it leads to two different structures because they're not exactly in the same place. But again, the formula stays the same. So these are known as optical isomers. So there's stereo isomers, there are structural isomers, and there are optical isomers or enantiomers. So these are not going to form mirror images. It's going to be obvious that they are different. So make sure that you know it's because we have asymmetrical carbons. They can be called enantiomers or optical isomers. And they are non-superimposable mirror images. If I lay them one on top of the next, they will not line up exactly. Okay, so now we're going to start talking a little bit about how we can build or break down these macromolecules. So this is the concept of metabolism. So when you hear people talking about, oh, my metabolic rate is slow and da-da-da-da-da, at the basis, this is what they're talking about. So metabolism, I know here it says removing water. They're talking specifically about the image we're looking at. But metabolism is just all of the chemical reactions that happen in your body. And when there are two major ones that deal with the building or the breaking of organic compounds. So when water is removed, that process is known as dehydration synthesis. If you think of what dehydrate means, it means to remove water. Synthesis means to make. So I'm going to make something, I'm going to connect something by removing water. You may also see this reaction referred to as a condensation reaction. And if you think of condensation as in the condensation of liquid on a glass, water on a glass that's cold, then again, we're pulling water off, okay? So in dehydration synthesis, we have our two carbon-based molecules, and they have on their last carbons, now notice these guys are going to be in ring structures, they're not chains, but on this carbon right here and this carbon right here, they have an OH group and an OH group. So if we get those two close enough to each other, the hydrogen gets attracted to this oxygen, which gives us H2O, and that H2O literally gets pulled off of the molecules that they are currently attached to. So this leaves space because this oxygen is still going to want to be bonded. This carbon still needs to form another bond, so they're naturally going to bond to each other. And if you look at the bottom part of the picture, you see how they arrange. Now, I know my picture is a little bit different to yours, but it shows you the exact same concept. So by pulling off this water, I create space, I create need to continue bonding, and now this oxygen gets bonded to this carbon right here, and these two molecules are now linked together. So the more times we do that, the more links we create in the chain. So this is how we build a biomolecule. So now the opposite, when we add water, we have the opposite effect. So this process of adding water is actually going to break that bond, And now our molecules that were linked together are now going to be separated from each other. And that process is known as hydrolysis. So hydrolysis splits up macromolecules, and condensation reactions or dehydration synthesis reactions build macromolecules. Okay, so for a quick breakdown of the word, hydro, of course, means water. Lyse means to break or to split. So hydrolysis is the breaking up of macromolecules by the addition of water. So if we look at our molecule here, this entire thing is connected, okay? Notice I have my oxygen that I'm sharing between the two molecules, so this bond is how they are linked together. So I'm going to add water, and by adding water, I do the opposite of dehydration synthesis. Adding water is going to take one of the hydrogens and attach it to that oxygen, which makes it stable. So now it's no longer interested in bonding with the other glucose molecule. And then the remaining OH is going to be added to the carbon on that glucose molecule. So now we've gone back to having two separate molecules. Okay, so now we can get into talking about macromolecules or large molecules, because macro means large. So when we say macromolecules, we are referring to carbs, lipids, proteins, and nucleic acids. And they are sometimes also referred to as polymers. And a polymer is just a large molecule that's built from the same subunit, the same smaller molecule, repeated over and over again. So we call the small subunits monomers, and we call what they build a polymer. Now those polymers are going to be held together in a chain by covalent bonds, which is why we started off this unit by talking about chemistry. So remember, those covalent bonds are those strong bonds that share electrons between two atoms. So like I mentioned, polymers are built from monomers. We take all of these small subunits and we assemble them together. Kind of want you to think of Legos, okay? If I take the same kind of Lego and I build a giant wall of Legos, the one single Lego was my monomer, the whole chain of Legos or the wall of Legos would be my polymer. So that process is known as polymerization, and it happens, so again, polymerization is just the assembly of a large molecule using the same small repeating molecules, the same small repeating subunits. And it's done through dehydration synthesis reactions, through the removal of water. Now, the opposite reaction causes the breakdown of polymers. So we go from polymers back into the individual units, and that is known as hydrolysis, okay, the adding of water. All right, so this is how we build and break macromolecules. Okay, so technically there should be a chart. You probably still have this chart on your slides. After the entire video is done, I would use that chart to go through and kind of hit the big items of all of these macromolecules. So just fill out the information for each of those biomolecules. And this is a quick little study guide where you can see a lot of information about each individual biomolecule in one location. Okay, so let's start talking about the actual biomolecules, starting with carbohydrates, which many people just refer to as carbs. So carbohydrates are the most abundant macromolecule that we have in life. And they come in a specific ratio, a specific format. They always are made up of the elements carbon, hydrogen, and oxygen in the following ratio. For every single carbon you will have 2 hydrogens, and you will have 1 oxygen. So it's what we call a 1 to 2 to 1 ratio. For every carbon, there are 2 hydrogens and a single oxygen. These guys, their most important job is to act as a fuel source for energy storage. But this is what we call quick energy. This is the energy your body is utilizing constantly all the time. We're not storing and holding this energy for long periods of time. You are using this daily. So if you are familiar with people who take pre-workouts or have a quick snack before, like a quick light snack, before hitting the gym, they are about to go and burn off all of the energy that's in that quick snack because they will be actively moving their bodies. So the only way your body is going to store carbs long term and it doesn't get stored as carbs is if you have excess. So what are some examples of carbohydrates? Well, pretty much lots of sweet things. So glucose, sucrose, fructose, for example, and then starch and glycogen are other examples. So think pasta, rice, potatoes, all crackers, all of those would be sources of carbs. Anything sugary, even if you're thinking of like fruit sugar, because apples are sweet, bananas are sweet, pineapples are sweet, all of them contain carbs in some form. So that's function number one. Function number two is to act as a structural molecule, to give support and structure to certain kinds of living things. And you see this a lot in plants, and you see this a lot in insects. So our examples here are cellulose and chitin, or chitin actually. So cellulose is actually found in the cell walls of plants and help to give that plant cell its very typical rectangular type structure, helps to hold it up. And chitin is found in the exoskeleton of arthropods. So think spiders or even like crabs. If you step on them, they make kind of like a crunch noise. Then that's where that sound is coming from. It's from the chitin in their exoskeletons. Okay, so if carbohydrate is the polymer, then what is the monomer of carbohydrates? Carbs are made up of monosaccharides. So mono means one, and saccharide actually means sugar. So literally one sugar. In terms of size, these usually have anywhere between 3 to 7 carbons, so they're not very big at all. They can also be found in chain structures like you're seeing here. So all of these are linear or chain structures. And then over here are the ring structures. So I know it says ring here on mine. It should say it in the right place on yours, hopefully. It's actually ring over here. These are the rings, and the others are our linear or chain forms. So carbohydrates also have the ability to form disaccharides. And di means two. And of course, saccharide means sugar. So disaccharides are two sugars that are linked together. Now, these could be two of the same sugars, or they could be two different sugars. It just depends on what we're making. So you guys are familiar with sucrose, which you know is table sugar, just the stuff you put in your coffee or your hot chocolate or your tea. And sucrose is actually a disaccharide. It's a combination of glucose and fructose. That's how we get sucrose. Lactose, which is known as milk sugar, so it's the sugar that's naturally in milk, is actually glucose plus galactose. And then maltose is malt sugar, and that is glucose plus glucose. So those are just some normal examples of, or everyday I should say, examples of disaccharides. So again, remember those are two or more saccharides that are linked together. They are formed from dehydration synthesis, and the bond that actually holds them in place is called a glycosidic bond. So that's still a covalent bond, but because we're talking about this specific covalent bond in carbohydrates, we now change the name to glycosidic bond. That's something you're going to realize about biologists. We love to name the same thing different things. I don't know why we do that, but we do. So right here, that's going to be your glycosidic bond. Right here, that would be your glycosidic bond. So up next in the hierarchy, if you will, are the polysaccharides. So the word poly means many. Saccharide still means sugar. So polysaccharides are many sugars linked together. So this is the actual macromolecule. This is the product of a polymerization where I'm putting lots of these monosaccharide units together. Okay. So as far as what polysaccharides do, it depends on if they are developed for energy storage or if they're developed for structural support. So if we're using them for energy, then plants tend to use starch. That's how they store extra glucose, is in the form of starch. So think potatoes, okay? Even if you eat carrots, for example, carrots are pretty sweet because that's extra glucose being stored in that carrot root. Animals don't use starch. Starch is a big molecule. It takes up too much space. So instead, animals use glycogen to store extra glucose. Now, if you are going to be used for structural purposes, plants are going to use cellulose. If you've ever eaten celery, you know those hard ribs, the long, skinny pieces that when you bite into the celery, sometimes that's the part you don't bite through, and you have to chew and chew and chew, and it still doesn't break up? That is your cellulose, okay? So cellulose is really strong, really long fibers that hold the plant upright. And then another example of structural polysaccharides would be chitin. And chitin is found in those arthropods, like I mentioned before, but they're also found in mushrooms. Okay, so let's go back to those storage molecules, of those storage polysaccharide molecules. So like I said before, starch is used by plants to store energy. And plants can store carbohydrates in two main ways. So the first type is called amylose, and it is a chain and it is unbranched. It means that the chain is just connected in one direction. So if you look at this picture, see how all the molecules are connected side by side? That would be an example of an unbranched linear chain. The other way they can store polysaccharides in the form of starch is through amylopectin. And amylopectin is branched. That's the major difference between these two. And I'll show you what branch looks like on the next picture. So animals, like I mentioned, stores their extra polysaccharides as glycogen. They don't use it, they don't store it as starch. And usually it's in the liver, and sometimes it can be in muscle tissues as well. But because your body reaches for carbs to fuel itself every single day, every second of the day, the polysaccharides need to be reachable, for lack of a better term. They need to be accessible so that your body can use it. So this is what branched means. So if you look at this picture, and you actually see it a little bit better here and here. So here you can see that they're all like connected to each other, but no one's changing direction until we get to here, and now they've changed direction. They're still connected, but I now have two pieces coming off of, whereas before I only had one. So if we look at this straight, here are my pieces all connected, but when I get to this carbon has an additional branch that starts above it. Okay, so I have multiple pieces all connected. Okay, back to our structural polysaccharides. So like I mentioned before, cellulose is made by plants as a structural fiber. It's found in their cell walls. And they are unbranched chains of glucose, which makes sense with my celery example, right? Cellulose is not something that our bodies can digest. So it gets lumped into the category of fiber when we talk about nutrition. So it's indigestible by most animals. There are some exceptions. So termites and any of your ruminant mammals, so cows, goats, sheep, any of the ones that have multiple stomachs, can have microorganisms that live in their digestive tract that actually have the ability to break down cellulose. So chitin is our next example of a structural polysaccharide, and it is really tough material, very resistant to weathering, very resistant to changes in temperature, the environment, that kind of thing. So it makes it perfect for being used for structural purposes for things like fungi and for the exoskeletons of arthropods. So when you look at it, it looks very similar to cellulose. But the main difference is that chitin's glucose molecules have these nitrogen groups in them. And it's that nitrogen group that gives it some of these unique properties of being so resilient and so strong. So that's why when you step on insects, sometimes you get this crunching noise. because of that extra chitin, that extra material. All right, so up next are lipids. And lipids are kind of unique in this world of macromolecules. They are going to be mostly nonpolar, and they are water insoluble. If you put them in water, they will not mix. They will float on the top. And it's because they contain lots and lots of hydrocarbons. And hydrocarbons are really stable. They have no need to react, so they don't. So there are three major categories of lipids, and they are neutral lipids, phospholipids, and steroids. So lipids are important for energy storage. So these are like our fats and oils that we tend to think about when we're thinking of lipids. Phospholipids provide the structure for biological membranes, so like our cell membrane. And steroids help the membrane structure to help the membrane structure to hold itself together, and they can also act as hormones. Okay, so starting with neutral lipids. Oh my goodness. Neutral lipids are nonpolar, as we've mentioned in the previous slide, and they have no charged groups at normal cell pH, which is about 7. So the most common example of a neutral lipid is called a triglyceride. So tri meaning 3, and that's going to give you a hint as to what they look like structurally. So triglycerides are used for energy storage, but this is going to be long-term energy storage. So remember me telling you that carbs are used for quick energy. When your body needs energy, it's reaching for carbohydrates, it's breaking down carbohydrates. But when you have extra, your body stores it in the form of fat, just in case you have long periods of time where food is not available to you. So that's why when you diet, or even when you exercise, or a combination of the two, it takes so much effort and it takes so long to actually break fat down. It's because your body doesn't like to break its fat down. It wants to hold on to its fat just in case. Think of it as your rainy day fund when it comes to energy. So why would we choose, and by we I mean the body, why would the body choose to store energy in fat and not in carbs? Well, it's because lipids can store twice the amount of energy as a carbohydrate. So just like with the other macromolecules, lipids are formed by dehydration synthesis. We're going to remove that water and create a bond. And it happens between two specific regions of the molecule. So the head region is what we call a glycerol molecule, and it's made up of 3 carbons. It's a 3-carbon alcohol. It's made up of an alcohol with 3 carbons. So that would be this region here. The rest of the molecule are known as fatty acid tails. And remember, this is called a triglyceride because there are 3 fatty acid tails. So that would be these guys here that I'm now outlining in blue for you. So my glycerol head is up at the top, and I have these 3 fatty acid tails. I tell my high schoolers that it looks like the letter E. A triglyceride looks like a letter E, which is really long tails. And the bond that holds them together, the bond connecting the carbon in the alcohol to the fatty acid tails, is called an ester bond. And I'll show you where it shows up. Hopefully it'll highlight. So right here, here, and here, that's our ester bond. Okay, so ester bonds are covalent bonds, but because we find them in lipids, we refer to them as an ester bond. So remember, in carbs, that covalent bond that holds your carbohydrate molecules together are referred to as a glycosidic linkage. Still a covalent bond, but we just name them differently depending on the macromolecule that they are a part of. Okay, so let's talk about some more characteristics of fats. So fats are semi-solid at biological temperatures, so think body temperature here. And fats are an example of a saturated molecule. So their fatty acid chains every single carbon is bonded as many times as it possibly can. So it's saturated fatty acids. They can be pretty long, anywhere between 14 to 22 carbons, and they usually only have a single bond between the carbons. So no double bonds, no triple bonds, okay? They are carrying the maximum amount of hydrogen ions, which is why we use the term saturated. They are full of hydrogens. And the reason they can carry the max number of hydrogens is because they only have single bonds between their carbons. Remember, carbon bonds up to 4 times. So the picture here is just an example of a saturated fatty acid chain. It's called stearic acid. Now let's talk about oils. Oils also fall under the category of neutral lipids, but they tend to be liquid at biological temperatures. So think of your seed oils. Think of olive oil, think of coconut oil, not coconut, sorry, but think of sunflower oil, vegetable oil. All of them are liquid at biological temperatures, at normal body temperature. These guys are unsaturated, so they have unsaturated fatty acid chains. What does this mean? It means that they don't just contain single bonds. They contain one or more double bonds. How many double bonds depends on the identity of the molecule. Because of those double bonds, they are not going to be maxed out with hydrogens. They'll have fewer hydrogens per carbon than they technically can carry, so we consider them unsaturated. Now, because of the presence of the double bond, it results in a bend or a kink in these fatty acids. And it always happens at the location of the double bond. So the more double bonds, the more bends or kinks you can expect. So they don't lay nice and straight like the molecule I showed you before, which was stearic acid. This one has a bend. It's at an angle, if you will. And this example is of oleic acid. So triglycerides are really useful when it comes to storing energy. They're efficient, let's say. And because of this, they can store twice as much energy per weight as it would take a carbohydrate to do the same thing. So for many reasons, animals choose to store their extra energy, their extra fuel, as fat, rather than the carbohydrate equivalent, which would be glycogen. Glycogen would actually take up more space and would be heavier to carry around. So as much as we hate our fat sometimes, our fat is there for a purpose, and it's actually the better of the two options that we have. So we always describe fats as long-term energy storage, twice the amount of energy as compared to a carb. It's just a really good way for you to store energy. Now, along with just using it to store extra energy, a lot of triglycerides have evolved for other purposes. So for example, if you've ever seen a bird, like a duck, preen its feathers, when they're like picking at them and like laying them all smooth, they actually have an oil gland at the base of their tail that's filled with a triglyceride specific type fat. And they use that to make their feathers water repellent. So any bird that lives in the water, think penguins, think ducks, think mallards, they are going to be really good at this technique where they keep their feathers water resistant to allow them to have the lifestyle that they have. So along with fats and oils, there's another category of waxes that still falls under the neutral lipid umbrella, if you will, that are called waxes. And waxes are fatty acids that have been combined with long chains of alcohols, those OH groups. Or they can also be combined with hydrocarbons, and they form insoluble wax. So think beeswax. Or if you are a plant enthusiast, maybe you've seen those plants where the tops of their leaves are very shiny. Well, that shiny coating is called a cuticle, and it's based in wax. It's a wax-based material. So honeybees, as you know, use wax to build their combs. Plants, like I said, have this covering called a cuticle that is made of wax, and it's a protective layer to prevent the plant from drying out. It prevents or reduces, I should say, the amount of water that that plant can lose based on its environment. Okay, so we are done with the neutral fats, and now we are moving on to another category of lipids called phospholipids. Phospholipids are a huge part of biology, mostly because they provide the structure and the frame for our biological membranes. So when I say biological membranes, I want you to think the cell or plasma membranes that we have to make up our cells. These membranes pretty much say, hey, everything inside of me is the cell and everything outside of me is not. So these phospholipids have a duality, a dual nature, if you will. They are amphipathic, and that means that they have regions that hate water, which we call hydrophobic regions, and they also have regions that like water, which we call hydrophilic regions. So you've seen the words polar and nonpolar as it compares to liking or fearing water. So now we're adding in the words hydrophilic and hydrophobic, which mean the same thing. So if you are polar, you are hydrophilic, you love water. If you are nonpolar, you are hydrophobic, think of phobia as in fear, you fear water. You want to get away from water. Excuse me. So when I take a phospholipid and I put it in an environment that's completely polar, like water, for example, these lipids tend to bunch together and cluster together in a very specific arrangement. They form what we call bilayers. Two layers with the polar groups on the inside, with the polar groups facing the water, and the nonpolar groups, the fatty acid tails, facing the inside, trying to get as far away from that water as possible. So the attraction and repulsion of water actually creates a really strong surface. I know we're used to thinking of cell membranes as just that kind of circular line that we draw around the cell, so it looks pretty flimsy, but these bilayers are actually pretty sturdy and they can hold up to a lot. Okay, so again, due to how this got imported, I don't have this image, but what you should be seeing on your note sheet, like on your PowerPoint, you should have 4 different images, and they just show you the regions of the phospholipid. And when I get to a picture that has that, I will go over these regions for you. Pay attention to the fact that if I am part of the top of the molecule or the head region, I am polar. If I am part of the tail region, then I am nonpolar. Okay, so let's spend some time going over the regions of a phospholipid. So we generally talk about the head region, which is all of this section, and it is polar. And then we also talk about the tail region, all of this section, and they are nonpolar. So remember, we can also use the word hydrophobic to refer to the tails because they don't like water, and hydrophilic to refer to the head because it does like water. One of the ways to tell the difference is the tails look like tails. They almost look like long legs, if you will. And the head region has the phosphate in it, and that's where the name phospholipid comes from. So now it goes on to show you some examples of what these frozen bilayers look like. So when placed in water, the layers are actually not stationary. The tails kind of move. The molecules themselves are capable of twisting and turning and flipping around and exchanging places with each other. So they're constantly in motion. Now, if I were to freeze the water, then notice how all of the tails are nice and straight, whereas in liquid water, you can tell that there is movement there. And then if you were to cut the layer in half, this is what it would look like. That layer is a 3D object. We always draw it like it's 2D. Here's this circle, but it's actually fully encompassing. And notice that the middle of the layer is completely empty. So it makes perfect sense why these layers are great to form the boundary of cells. Okay, so remember there is a section of our bright space, eCampus, if you will, where I have resources for you, and that's where these podcasts, these extra videos, and that kind of thing, at least links to them, can be found, as long as I could still find working links. Some of these videos are older, but for the most part, the links tend to work. So these are great ways to revisit information, to study, to brush up your memory, and to make sure that you have an additional explanation for understanding. Okay, the last of the lipids are known as steroids, or sometimes they're called sterols with an OL at the end. So steroids are lipids, but their structures are very different. Their structures are based on rings as opposed to tails. So if you look at the image, I'm going to switch to my laser to point this out. You have these fused rings, 1, 2, 3, and 4. And yes, I know this one doesn't really look the same. It's not a hexagon, but we still consider it a ring structure, okay? So this is the shape. Whoops, I'm so sorry, y'all. This is the shape that your steroids take. They don't look like these long chains of fatty acid tails that we've kind of gotten used to. And all steroids have these 4 rings. So in this one, there's 1, 2, 3, 4. This one over here, 1, 2, 3, 4. What you would notice is some of my molecules are different. So like here I have this methane group, no methane group here. Here I have an alcohol group, no alcohol group here. I have a double bond, an O double bonded to that carbon instead. You'd also notice that here there is an extra line. That extra line just means double bond. So none of these rings have double bonds. This ring has a double bond. But over on this new steroid, this ring has 3 double bonds. So like there's some little tiny differences, but they make all the difference, literally, because the reason this guy is one kind of steroid and this guy is a different kind of steroid has everything to do with what extra groups and how many double bonds are found on those rings. So the most common steroid for animals is cholesterol. And for plants, they have steroids too. They're called phytosterols. And both cholesterol and phytosterols have the ability to change the behavior and the characteristics of these plasma membranes, of these bilayers that they can be a part of. Now, steroid hormones, so lipid-based hormones, are also really useful as regulatory molecules. They can help to bring information and signals throughout the body. So they behave a lot like hormones, if you will. So I know you can see the names on your image. So this is estrogen, what we consider, or estradiol, a type of estrogen that we consider the female hormone. And then this hormone is testosterone. So again, they look very similar, but the absence of this and the changing of this to an OH makes one of them estrogen and the other one testosterone. And then these are going to be some of the reasons that our female duck looks the way she does and our male duck looks the way he does. So as I'm sure you are aware, there is a correlation between fats, cholesterol, and coronary artery disease, or different types of heart disease. So what you're looking at here in terms of the picture, is the coronary artery, and these really bright yellow areas are what we call plaque, arteriosclerotic plaque. So pretty much fat deposits that have built up in these arteries. And when those fat deposits build up in the arteries like this, it actually makes the arteries smaller. So this is what I need you to envision. So here is my artery. Arteries have muscles in them. Okay, they're double lined, and this one's nice and open, and blood can flow in and flow out, and there's no issue. Here is an artery that has plaque. Okay, so see how the opening is a whole lot smaller? That tells me right away that the plaque is pushing into the artery, and it also tells me that the artery has condensed in size. The opening is smaller. So that means that this vessel will carry less blood, which means your heart has to work even harder to get the blood through it because it doesn't hold as much as it once did. Okay? So if you've been to the doctor recently or not, no judgment here, but if you haven't, make an appointment. And your doctor has tested your cholesterol level. One of the things they normally talk to you about are your levels of HDL and your levels of LDL. So HDL stands for high density lipoprotein, which is very often referred to as the good cholesterol. So this is the cholesterol that's going to absorb cholesterol in the blood from your food, literally, take it back to the liver, and then the liver is going to flush it out of your body when it goes through its detox process. HDLs do not cause coronary artery disease. Okay? They don't stick to the walls of your arteries. Now, LDLs, on the other hand, which we call low density lipoprotein, is one of the groups of lipoproteins that will cause plaque. It is the bad cholesterol, if you will. Okay? Your body doesn't flush this as easily. It gathers around in parts of the body and then creates these areas or these pockets where it's making it harder for your heart to do the job that it needs to do. The third category of macromolecules are the proteins. The proteins are going to be the most expansive of the group because they perform so many vital options in living things. So just know, a couple of years ago, when apps, I might be aging myself, but before apps became really, really mainstream, and people were realizing, we can make a new app to do a bunch of different things that didn't exist before. The saying, there's an app for that, kind of came out and was a thing for a while. Well, think of this as, there's a protein for that. Everything you can think of, not everything, but lots of things that the body does, proteins are the ones doing all the heavy lifting. So proteins perform lots of different functions in your living systems. They help with structure. They help with transport. They help with communication. And the really big ticket item are enzymes, which is a whole nother unit unto itself. So what are the elements found in proteins? They are carbon, hydrogen, oxygen, and nitrogen. So the first three we know we find in carbs and also in lipids, but nitrogen is a new one. The monomer or subunit, of a protein molecule is called an amino acid, and there are only 20 of them total on Earth. So they're pretty rare, it's one way to think about it. So when I put a bunch of amino acids together, the bond that holds them in place is a peptide bond. And the molecule or the polymer that they make are polypeptides or proteins. So when you think of the word polypeptide, it literally means many peptide bonds. Okay, so this table just goes over some of the functions that proteins can have in the body. So proteins can be structural, where they're being used for support. Okay, so for example, in muscle fibers, you have proteins there. In cell walls or even cell membranes of your plant and animal cells, you will have proteins that are designed just to hold the cell in place and hold its shape together. We also have enzyme proteins, also called enzymatic proteins, that help to increase the rate at which a reaction is going to take place. We'll have a whole unit over this, so don't even worry about it. We have transport proteins that help things to enter and exit the cell. Again, there's a whole nother unit over this coming. And then we have modal proteins, proteins that allow cells to actually produce movement. They carry things, they move things, they allow things to split apart. So you see them a lot in processes like cell division, okay, or even the movements of some of the organelles within the cell itself. Okay, so to continue from the previous slide, we have regulatory proteins, and it's their job to either promote or increase the activity of other cellular molecules, or sometimes it's their job to stop or inhibit the activity of cellular molecules. There are receptor proteins, which pretty much behave like your cell phone or your antenna, if you will. It's their job to intercept molecules and to trigger some kind of response from the message that those molecules send. There are hormonal proteins, which carry signals between cells, defensive proteins that help your body to fight off pathogens or anything that can cause you to be sick, so think antibodies here. And then there's storage proteins, which help to hold substances like amino acids in a stored form. So if you are a fan of egg whites, that would be an example of a storage protein. Albumin is a storage protein for eggs. Okay, we have a couple of different kinds. So let's talk about the monomers, or the building blocks of proteins. So proteins are built from amino acids. Amino acids are, oh, I'm so sorry. This should have been red. Amino acids are the building blocks of proteins. They are the monomers of proteins. And they're made up of the same 4 regions. And again, the images is distorted. It should be fine on your paper copy. So I'm going to redraw this in just a second. Okay, so thanks to Video Magic, there is what an amino acid looks like. So I'm going to color code the 4 sections and then color code the diagram, and you can do the same, or whatever method works for you based on what you have with you. So the first section of an amino acid is the amino functional group, or the amine functional group. Then we have the carboxylic acid functional group. Those are going to be found on either end of the molecule. We have a hydrogen that's bonded to a central carbon. And then we have what we call the R group, or a variable side group. So on the actual molecule, this is going to be my amino group here. My carboxyl group is over here. My single hydrogen on my central carbon is found right here. And my R group, or variable group, is found right there. Okay, so it's a nitrogen, carbon, carbon backbone, and then all of these things are attached to either the nitrogen, the central carbon, or the carbon to the right. So there are 20 amino acids, and the only difference between them is this R group that is found on the central carbon. The R groups are going to differ in terms of shape and size and charge, reactivity, how they behave in water, all of those things. So there will be a chart coming that shows all of the R groups, and the good news is you don't have to memorize those structures. The quote-unquote bad news is you do need to be able to recognize the names of the amino acids if they, not if, but when they pop up. All right, so here is that chart with those amino acids, and you can see that we can kind of break them up into certain groups based on the behavior of their R group. So we have all of the amino acids that have non-polar side chains. These guys are pretty stable. They're not going to react very much. They're happy where they are. Then we have the polar side chain groups, which will all react well in water. And then we have the electrically charged side groups. So these are the R groups that are either overall acidic or overall basic. Okay, so how do we build proteins? Well, we build them by connecting the amino acids using a covalent bond called a peptide bond. When that happens, when we start making 2, 3, 4 of these connections, we build what is known as a polypeptide, literally many peptide bonds. Okay, and then the reaction that is going to create that peptide bond is the same reaction we've been talking about for all the others. It's going to be dehydration synthesis or condensation, same thing. Okay, now note the peptide bond is located between the last C of, hold on, let me switch to my laser. So note the peptide bond is going to go between the last C of the first amino acid and the N of the second amino acid. So when it's completed, right here is where that peptide bond is. So here is my last C on amino acid number 1, and here is my N on amino acid number 2. Okay, now we use these terms N-terminal and C-terminal just to help us figure out where is the amino group and where is the carboxylate group. It gives us the direction of the molecule. The C-terminal is the end that the carboxylic group is on because it has that last carbon. Okay, so we will step through these in more detail in just a little bit. But the next thing we're going to start talking about are protein shapes and protein structure. So this is an overview slide, okay? So from this slide, we know that there are 4 levels of protein structure: primary, secondary, tertiary, quaternary. Here you have pictures to show you what those levels involve. So primary structure is just the order of the amino acids. Secondary structure is the same order, the original primary structure, but now we've added this twist or bend to the molecule. Tertiary structure looks like the molecule has become 3D fully for the first time, so the whole thing is folding on itself. And quaternary structure is the arrangement of the individual polypeptides that make up the protein and how they're all associated together so that they can do their job. It also gives you an idea of size. So your primary structure and secondary structure are based on the monomers, the individual amino acids. When we have a peptide, we've had less than 50 amino acids in place. When we have a polypeptide, we've joined more than 50 amino acids in place. Okay? So here where it says be able to distinguish the shapes from one another, if I gave you this image and I asked you what structure that was, I need you to know that it's primary. If I gave you this image and I said, what structure is this? You need to be able to tell me that it's quaternary. So that's what that side note means. Okay, so here you can see 2 computer-generated models of what a fully folded protein would look like. So in these regions where it just looks long and thin, that is going to be just primary structure that's happening here. In these regions where you can see that cylindrical shape, that tells me that secondary structure is happening here. How it's folded up is my tertiary structure. And here they've included those disulfide bridges. Notice that you don't have many of them. It's really just to hold one end of the molecule to the middle and the other end of the molecule to the middle. This diagram over here is what's known as a space-filling model. So they use different colors to represent different elements, but you get an idea of physically how much space, what's the outline, if you will. You don't see too many fine details, but you get a good idea of what the molecule looks like, or not the molecule, but what the protein, I should say, macromolecule, looks like when it's fully folded. Okay, so you will hear, hold on one second, me say this a lot, especially when it comes to proteins. Form and function, shape and behavior are key. If I lose my shape, I can't do the job I was designed to do. Okay? So the shape of your protein is going to help determine what exactly that protein does. Your shape determines your function. Okay, that's super important. What determines the shape then? Well, the type of protein, meaning the type of amino acids, the position of the amino acids, and the number of the amino acids will all help to determine how that particular polypeptide can be built. And how the polypeptides are built determines how they fit together, so the overall shape. Now, some of this has been cut off a little bit. Hopefully you can still see it on your printed copy. But proteins whose shape has been disrupted there's a word we use to describe that, and that word is denatured. Okay, so it means that it no longer has up to that quaternary level of structure. It's all loose and unfolded. It no longer has the proper shape, so it can no longer have the proper function. Okay, so what is going to cause my protein to lose all of these specific shapes that hold it together? The main culprit is normally heat. Sterilization can also cause this. UV radiation or irradiation can also cause proteins to denature. Really high salt concentrations and superacidic environments can also alter the shape of your protein. Okay, I apologize. I do not know what this random red arrow is in the middle of the slide, so just ignore that. So now we have moved on to the last class of macromolecules, and these are the nucleic acids. So there are only two examples of a nucleic acid. Nucleic acids are either going to be DNA or RNA, and their job involves being the information storage molecules of all living things. Okay, so our genetic information is stored in the form of nucleic acids, either DNA or RNA. So the elements found in your nucleic acids are carbon, hydrogen, and oxygen. We expected those. This one also has nitrogen, like protein does, but it also has an additional element in the form of phosphorus. Okay, so carbon, hydrogen, oxygen, nitrogen, and phosphorus are the elements in the nucleic acids. Nucleic acids are built from the monomers of nucleotides. So they're monomers called a nucleotide. And that nucleotide actually has three parts to it. It has a nitrogen base. It has a 5-carbon or pentose sugar. And it has one, if not more, phosphate groups. So we can draw a very simple sketch off to the side. Now that'll appear in just a second. Okay, so there's a very simple schematic of kind of the general arrangement of what that nucleotide is. So all three parts together make the nucleotide: the nitrogen base, the pentose sugar, and the phosphate group. Now, just like all the other macromolecules, these monomers are going to be held together by a covalent bond. Because these covalent bonds show up in nucleic acids, they have a different name. So they are going to be held They are going to be held together by a phosphodiester bond. And that is the name of the covalent bonds found in nucleic acids. So this slide does a great job of comparing the two types of nucleic acids, specifically DNA and RNA, on some general structure type things. So the first is that DNA is double-stranded. We normally refer to it as a double helix, whereas RNA is generally just a single-stranded molecule, so just one strand, whereas DNA has two. DNA on its pentose sugar is missing an oxygen. So if you look at this highlighted region, on this second carbon, we have a hydrogen group that doesn't have an oxygen. So as a result, we name this pentose sugar deoxyribose. So deoxy for no oxygen on my ribose sugar is how I think about it. Okay? Whereas on RNA, it looks very similar, but on that second carbon, I have a hydroxide group. So I have the oxygen. So this sugar is just known as ribose sugar. So the R in RNA stands for ribose, while the D in DNA stands for deoxyribose. And it's one word, okay? And it just talks about the fact that DNA is missing an oxygen on its second carbon, whereas RNA has that oxygen attached to a hydrogen on that second carbon. Then the other thing we can look at for differences are the nitrogen bases, the identity or the type of the nitrogen bases. So DNA uses the nitrogen bases thymine, cytosine, adenine, and guanine. We normally use the letters T, C, A, and G to represent them. So just the first letter of the name of the nitrogen base. And RNA has very similar bases with one exception. So RNA also uses cytosine, adenine, and guanine, but it has the addition of uracil. So there is no thymine in RNA. Thymine is replaced by uracil. Okay, so those are the three major differences between DNA and RNA, kind of in a nutshell. Okay, so this image just goes over the structural plan of a nucleotide. So like I mentioned before, that the nucleotide can be made up of multiple phosphates. That's a possibility. And there are reasons why a living system may need more than one phosphate, and we'll address those when we need to. So the version I drew for you just involved one phosphate, one sugar, one nitrogen base. This one just shows that you can have additional ones, and we'll come back to this, so don't stress. All right, so that first picture is very simplistic. This one shows more of the structure. And again, we're just paying attention to the three major parts, which are the nitrogen base, the sugar, and then the phosphate group, however many of them may be present. So DNA and RNA both have what we call directionality. And the directionality is a result of the carbons in that pentose sugar. So I know you have pictures on here that label all these things, but I'm going to show you this in real time. I'm going to draw it in real time, not like speed it up or pause it and then the image just appears. So we're only focused on the sugar for right now. So that pentose sugar actually only has 4 carbons in the ring itself. It starts off with an oxygen. Then we have a carbon here, carbon here, carbon here, and a carbon here. Okay, so where those lines intersect, that's where that carbon is. But you're probably thinking, but you kept saying 5 carbon ring or pentose. You're right, because the 5th carbon isn't part of the ring. It sits above carbon number 4. So that's where our 5 carbons come from. Now, we number these carbons in a specific order, starting with the carbon that is downstream and to the right, if you will, so this direction of the oxygen. That first carbon is known as carbon 1 prime. The second carbon is called carbon 2 prime. The third, carbon 3 prime. The fourth is carbon 4 prime, and that 5th and final carbon that's at the top is carbon 5 prime. So the 2 you need to pay attention to are carbon 3 and carbon 5. They tell the direction. Okay, so when we say up here that 5 prime and 3 prime designate direction, that is what we're talking about. We're talking about this carbon here and this carbon here. Why those 2 carbons? Because outside of carbon 1, which is where your base is attached, it's these 2 carbons that the other nucleotides will be attached to. Okay? So you will see this, and I'll explain this more when we need to, but you will see this again, and you'll also see this next note again. So DNA reads in the 3 to 5 prime direction, but it is synthesized or built in the 5 to 3 prime direction. And keeping that separate is going to be really important. So when I read DNA to make copies of it or to make proteins pretty much from my genes, I'm going to read I'm reading this way. I'm reading up, so from 3 to 5, if you will. But when I build DNA, I have to build from 5 to 3. Okay, so let's talk a little bit more about the nitrogen bases. So the bases can be broken down into 2 big groups. We can talk about the pyrimidines, and we can talk about the purines. The pyrimidines are made up of 1 carbon-nitrogen ring, and they are uracil, thymine, and cytosine, those 3. So you can see a picture of them over to the right. The purines are actually made up of 2 carbon-nitrogen rings that are fused together, so they're attached on the sides, and those purines are going to be adenine and guanine. So I don't really have a super cool way for you to remember pyrimidines, but this is how I learned to remember purines. Adenine is A, guanine is G. So their initials are Ag. And Ag on the periodic table is the element silver. So to remember purines, I think of the phrase pure silver. And that's how I remember that A and G, adenine and guanine, are the purines, which means everybody else is a pyrimidine. I hope that helps you. Okay, so this is going to come back a little bit to the directionality that we talked about before. So when I'm building DNA and RNA chains, I have to produce a link. I have to make a covalent bond between nucleotide 1, 2, 3, 4, etc., etc., etc. Okay? Now, my phosphate group sits on carbon 5 prime. So when I link a new nucleotide beneath that, so let me use my laser here. Actually, before I do that, let's do this. Okay, so you may have noticed that I colored in the nucleotides on the RNA side. I only did it on the RNA side because it's just one strand, so it doesn't get as confusing with all the different images as on the DNA strand. Know that it works the exact same way for DNA. So now I'm going to switch to my laser pointer. So this entire thing, my phosphate, my sugar, my base, that is one nucleotide. When I build this molecule, I have to attach all these others, right? So look at where the attachment goes. Here's my carbon 3 prime. That carbon 3 prime is going to get linked to the phosphate of the next nucleotide. And then that nucleotide is attached. Then on that second nucleotide, its carbon 3 prime gets linked to the phosphate of the third nucleotide. So now that one's attached. Okay, and we just keep going until they're all linked up. So that line between the carbons and the phosphate, so the carbons of the first nucleotide and the phosphates of the second, third, fourth, however many nucleotides, that is your covalent bond that we call a phosphodiester bond. Okay, so the phosphodiester bond goes between the carbon and the phosphates. And that's what it's supposed to be showing you over here. Okay, so like we said before, these nucleotides are held together by phosphodiester bonds. And now let's switch back to DNA. Something I need you to notice is that DNA doesn't run in the same direction. So here's strand number 1. Pay particular attention to the position of the carbon. And now here's strand number 2. Well, now the carbon looks like it's flipped upside down, doesn't it? Because it is. And the reason that has to happen is because these nitrogen bases have to line up in the middle. And they have to be held together by hydrogen bonds. So let's write ourselves a note about that. See why it was so important to learn about chemistry and bonding before we started learning about biology? Biology is based in chemistry, so you can't do one without the other. So because of where the nitrogen bases have to line up, and because of how they line up, one of my strands has to run in the opposite direction. So see here how I start with 5 prime? Notice here I'm starting with 3 prime. So 5, 3, 5, 3, 5, 3, 5, 3, if we're looking at the strand on the left. But if we're looking at the strand on the right, it goes 3, 5, 3, 5, 3, 5, 3, 5. So the term we use to represent that is the term anti-parallel. We say that the DNA molecule or the DNA strands are anti-parallel to each other. They run in opposite directions. Now, these nitrogen bases don't just pair up however they feel like pairing up. They have to pair up in a specific pattern, and they are held together by the hydrogen bonds between them. Okay, so we use the term base pairing rules to describe how they pair up in DNA and in RNA. Now in DNA, A and T like each other, so they pair up. And note they're held together by a double bond. Okay, so you can see that over here. Here's hydrogen bond number 1. Here's hydrogen bond number 2, double bond. Okay, A and T. It's the same for C and G, except C and G are held together by a triple bond. But they like each other, so they always pair with each other. All right, and then you can see that over here. So 1 hydrogen, 2 hydrogen, 3 hydrogen bonds. Now RNA, C and G, again, still like each other, still held together by a triple bond, just like in DNA. But remember, in RNA, there is no T. There is instead U. So A and U, adenine and uracil, will pair up in RNA, and they too are held together by a double bond. Another good way to think of the base pairing rules is that you have to have a mixture of the 2 types of nitrogen bases. So purine is always going to bond to a pyrimidine. It's not going to be 2 purines or 2 pyrimidines. It's always a purine to a pyrimidine. So because of this base pairing, the nucleotide sequence, the order of the A's and T's and C's and G's on one DNA strand is going to be complementary. It matches up. It complements the other strand. Now, RNA isn't going to have this necessarily, at least not in this format, because it's single-stranded. But DNA has complementary strands. They match together. They fit perfectly together. They were meant to be together, if you will. And it's because of this that we can use DNA to store our genetic material, because we're going to be able to code DNA. But we will talk more about that later on. All right. I know this one was long, but this is it for the macromolecules video, and hopefully it was all good. If you have questions, please do not hesitate to reach out. Bye, guys.